Hello, everyone, and thank you for joining us today. I'm Misty from the Dravet Syndrome Foundation, and today we have special guest, Dr. Barry Tico. He's the Chief Medical Officer at Stoke Therapeutics. Just a few little things to let you know. Please, when you come in, just mute yourselves and hold all questions until the end. So Dr. Tico will be taking questions at the end. You can either put them in the chat box. If you kind of hover down below, you'll see that chat box. You can type them in there and I'm happy to read them for you. Or you can take yourself off mute afterwards and ask those questions live either way. So I am going to go ahead and hand it over to you, Dr. Tico.
Thank you, Misty, and thank you, Jenny, and thanks everyone for joining this webinar. And of course, I wish we could do it all in person, but for now, this is the next best. And happy for those of you who are on video to be able to see you. As Misty mentioned, I will be taking some questions after the presentation, but I'll start with a few slides just to give you some information about our program and our potential medicine for people with Dravet syndrome. So let me just get this going here. First thing I have to say is there are some forward-looking statements in this presentation. And the first thing I know is many of you have questions. There are many questions we've gotten, and Misty's gotten, and Jenny and others. Nicole has answered a lot of questions, Marianne. So we'll go through some of these questions and try to give you some answers. So who are you? Who is Stoke? What is TANGO? What does STK-001 do? Why is this a possible treatment for children with Dravet syndrome? Very important question. Can you tell me about the butterfly and MONARCH studies? When is the testing of the medicine for Dravet syndrome starting? And where can I sign up my child for testing? So hopefully all of those answers will be given to you during this presentation. And if you have others left at the end, then we'll go through those as well. Oops, sorry. So who am I? I'm the Chief Medical Officer at Stoke Therapeutics. Give you a little bit of background information. I was born in Chicago and I live now just outside of Boston. My job at Stoke is to plan for clinical trials with our medicines, especially focusing on our medicine for Dravet syndrome, which we'll be talking about. I work with many researchers. I talk to families, just as we're doing today, and clinicians. And then I'm also working with a group of internal people who are helping with our clinical trials, and some of them are even on this call today. I like to go skiing and biking and running, and I read a lot. My biggest accomplishment was being married for 33 years. And my favorite quote that I tell others is, whoever saves one life saves the entire world. And that's part of why we're so dedicated to what we're doing here at Stoke. We hope to save many lives and improve many lives. And there on the left, you see my wife and daughter as well. So who is Stoke? Stoke is a company that's actually been around for 5 years. We're based in Boston. And we are a RNA medicines company. And I'll tell you a little bit about what that means in a little bit. We focus on severe genetic diseases. And we try to have a significant impact on people who have those diseases. We do that by trying to upregulate protein expression. And I'll explain all of that to you in a moment as we go forward. The basis for our medicines are something called TANGO. And it means targeted augmentation of nuclear gene output. And this is going to be a little bit technical, but I'll go through it. So we have the TANGO mechanism is a way to increase protein levels inside the cell. And that is a difficult thing to do, actually. But when we are able to do it, what we do is focus on what causes a disease and look for diseases where there is especially a special factor missing, protein missing. And then we can use our technology to increase the levels of that protein in the cell. And that allows us to then target especially diseases where a mutation in that disease can cause a protein, a special factor not to work or not to be present. And In this, we do it in a very specific way that targets specific cells in the body and can also only target the gene that we're looking at. So as we'll talk about with Dravet syndrome, there's the SCN1A gene, and our approach can target only that gene, be very, very specific in which cells and in which gene we can focus on. Our medicine is also can be given at different doses so that the effect that we want to have can be based on how much of the medicine we give. The more medicine we give, potentially the more of effect, but then at some point there can be a top level, a plateau, where giving more medicine doesn't help anymore. And we figure that out. We know exactly how much we want to give, and that helps us be a very precise medicine. And I won't focus on it, but the manufacturing of our medicine is relatively straightforward, so we don't have to spend a lot of time or effort trying to perfect that part of our medicine. So I'll show some pretty pictures here, which will go into a little bit of very technical detail on how exactly TANGO works. But on the slide here, you see, as many of you learned in biology, the DNA is our genes. It's what we carry from one cell to another and what one parent gives to a child. And in the DNA, there is a message there that tells the cell how to make certain proteins. These are the important building blocks of the cell. And in that message, there can be certain other additional signals in that message that get put in there that help the cell tell how much of this factor to make. Then, in addition, there's something that we call an NMD exon. It's a special coding part of the mRNA that's called a nonsense mediated decay exon. And that is a signal to the cell to get rid of that messenger RNA. So what you see on the right side of this figure here is when that little blue thing is there still in the RNA, the cell breaks down the message. It does not make the protein or the factor, special factor from that. When that blue thing is gone, as you see on the left side there, then that does not get broken down by the cell, and the message then can make the protein that it's supposed to. So what our medicine does here, and you see that in the fancy red comb there, our medicine can find that destroy me signal that's present in the message and remove that destroy me signal. And then as a result, as you see there, that blue thing is gone now on both sides. So now there's more of the message that the cell can use to make more of the protein. So that's the basis for our whole approach, is we use a small chemical to remove the destroy me signal from the message, and then the cell can make more protein inside the cell, which is, as I said, the goal for us, but not always easy to achieve. So I'll show it to you in a different way, because this is a difficult thing to understand. It took me quite a while when I was first joining the company. So I'll show it here in the context of a disease such as Dravet syndrome. And that is what we call a haploinsufficiency disease. What that means is the DNA, which comes in 2 different strands in everybody, in children and people with Dravet syndrome, one strand is normal, or is what we call wild type, and the other strand has the mutation. So here we show that in sort of orange and red. The orange one is the regular, normal form of the gene, and in the red there you see a mutation. In the case of Dravet syndrome, it would be a mutation in the SCN1A gene, which causes the protein not to be made from that gene. Now, it happens in the SCN1A gene, and in about a third of all genes actually that are present in the body, there is this NMD signal, this destroy me signal. It's shown here in that gray bar. So as I mentioned, when that gray bar is present there, as you see by that disappearing piece of messenger RNA, both in the orange and the red one, that goes on not to make any protein. And only the one that has the gray bar missing goes on to make protein. Now, in the case of Dravet syndrome, in addition, the orange is making the regular copy of the sodium channel, and you see that on the cell surface there. But the red one has the mutation so that it does not make that protein, and it does not make the sodium channel component. And that's what's causing all of the disease problems in children with Dravet syndrome. So here's the next thing. When we put our, in this case, green antisense oligonucleotide, we call it, which is binding specifically to that destroy me signal. Our green medicine removes that destroy me signal, as I showed you before. That makes more of the message. It makes more of the mutant message, but because that's not having any effect in the cell, then there is no response to that. But it's making more of the wild type message as well. That's making more of the protein to put it onto the cell surface. And so now there are more of these sodium channels. And the goal is to restore it back up to the normal level that is present in the cell normally, in the nerve cells especially. And so that's how we aim to improve the disease for children with Dravet syndrome, is to take what is normally only half the normal amount of these sodium channels on nerve cell surfaces and bring it back up, restore it back up to the normal levels. And we've been able to show that we can do that now in many ways. So just to summarize here, what is STK-001? It's a new medicine for children and adults with Dravet syndrome. It's an antisense oligonucleotide, and what that means is it is a small chemical that can bind specifically to a single site on that messenger RNA and bind very tightly. And as I showed you, it causes the destroy me signal to be removed. It's not gene therapy. So you may have heard of gene therapy. What gene therapy is taking the whole gene that would be coding for, in this case, NAV1.1, the protein that causes the protein mutation that causes Dravet syndrome, and that would be putting that whole gene back into the cells. It's very difficult to do. What our approach does is just work on the messenger RNA level. So it's not, the gene therapy typically works at the DNA level. Our approach works on the RNA level. And that gives us special properties that allow us to, as I said, focus only on one specific part of the RNA and to focus only on certain cells and be able to increase it or decrease the amount of protein from that RNA by adjusting the amount of medicine we give. So our medicine is designed to increase the NAV1.1 protein. This is that sodium channel that I mentioned to you that comes from the SCN1A gene. And it's designed to, in this case, restore it by doubling it back up to the normal levels in the brain. And that, as I mentioned, will potentially correct for the cell problem that is present in people with Dravet syndrome. Our medicine is given directly to the brain. So we don't give it to the whole body, we give it right into the spinal fluid, which is the fluid that goes around the brain. And that procedure is called intrathecal. It means that there is a small needle that has to be put into the back around the spine, and the medicine gets directly injected into the fluid. That allows us to give a very small amount of medicine, because since it's only going into the spinal fluid and it's only going to the brain, we can give a small amount. And as I'll show you in a little while, this is something that we think will be given not very often. So maybe even only every 4 or every 6 months, so twice a year or maybe 3 times a year, this injection would have to happen. And it would have to be a medicine that is given lifelong, though, in order to be able to continue to have its effect. And we do have quite a bit of animal data now that show that everything I was showing you about being able to increase the NAV1.1 levels does actually happen in animals, and gives us quite a bit of confidence that it will happen in people as well. So I'm not going to go through the animal data, actually, because Laurie Isom, who some of you may know, and is a very important contributor to the Dravet Syndrome Foundation, is going to give her own talk on the work that she has done at the University of Michigan with our medicine. And she's generated with her group and in collaboration with our scientists, some very, very nice data to support some of the claims that I've made here. I'm just going to give a quick summary of these, because I don't want to steal Lori's story, but I encourage you, when the announcement goes out for the date for her presentation, that you all join that one just as you join this one and get to hear more about what the potential for the medicine is. Excuse me. So what we have demonstrated is that in a mouse model for Dravet syndrome, so this is a mouse that was designed to have a very similar genetic structure to children with Dravet syndrome, and has a similar effect that the NAV1.1, the sodium channel special protein that is lower than normal in people with Dravet syndrome, is also lower than normal in the Dravet mouse. And we showed that with a single injection of STK-001 into the mice, that protein that is missing by half is now restored back up to its normal level. That for us was the best news that we could have, because that tells us that we are actually able potentially to take the nerve cells that are not functioning well and not communicating well because the sodium channel is not present at full level there, and we can perhaps make that sodium channel go back to its fully normal level. As a result, in this mouse model, the mice normally with this disease have seizures and die early on. And what we were able to show with our injection was that the mice had a significant reduction in their seizures, and many, many, many more of the mice lived than would normally have been expected. So that tells us that these injections that are causing the NAV1.1 to go back to normal are having an effect in terms of being able to prevent seizures and death in these mice. We've also looked in monkeys, who are, as you know, are very similar to humans. We've looked with a similar way where we do an intrathecal injection in the monkeys, and we show by looking in the brains of the monkeys that our medicine is going to most parts of the brain, and again having that similar effect where in the brain parts, these are now normal monkeys, these are not monkeys with Dravet syndrome, but even in these normal monkeys, we can show that the protein levels in those monkeys, in the brains of those monkeys, are now double what they normally would be. So that's exactly what we're aiming for, is just that doubling. And we have figured out how to give lower or more amounts of our medicine to titrate. That means to get just the amount of change in the NAV1.1 that we're looking for. So that, as I said, means we can adjust the dose, and we know from our safety results in our testing that we have a good range that we can use our medicine in, so that we can definitely give the medicine in the range that will have the effect in the brain that we are hoping for. We also showed in the mice that this effect of the changing in the NAV1.1 levels can last a long time. In this case, almost 4 months in this mouse model, and we also have some data in the monkeys that showed that the effects can last for 4 or even longer months. So that's why I told you that the medicine may only need to be given 2 or 3 times per year, because when the medicine goes into the cells, it lasts a long time. And so we think that it does not have to be given very often. And then the final thing is, when we gave it even to the monkeys at the levels that we think will be the same levels that we will need to have to give in people, and even at much higher levels, we did not see any significant safety concerns that would prevent us from moving into the testing in people. So that's a little bit about our medicine. Tell you a little bit about why we are focused on Dravet syndrome with our medicine, and what we think that we can bring special that other medicines for Dravet syndrome have not been able to do. And that's mostly focusing on, as you all know, the fact that Dravet syndrome is not just about seizures, it's about many other things. And it's about, in intellectual disability that children have, and the difficulty walking that they develop, and the speech problems, behavior problems, and the sleep problems. And the medicines that you're giving your children right now are mostly designed, as you know, to lower the amount of seizures. But they're not actually addressing the main cause of Dravet syndrome, which is what I've been talking about up till now, is this change in the NaV1.1 level. So the medicines that are given for Dravet syndrome right now do not, as far as we know, have a direct effect on the NaV1.1 level. So they're not really correcting the actual cause of the disease. And they also focus on just the seizures and trying to lower the seizures, and have not really focused on trying to change these other issues that all of you know are very important problems for people with Dravet syndrome. Our hope is that by changing the actual cause of disease and restoring these nerve cells back to their regular function, that we can not only lower the seizures, but also address many of these other problems that children and people with Dravet syndrome have. And the reason we believe that is because, as I said, our medicine is designed to actually try to correct the actual problem in the nerve cells that are causing these issues. I'm going to tell you now a little bit about some of the clinical trials, the 2 clinical trials that we have started as a company. And one is Butterfly, and the Butterfly is inspired by the Dravet Syndrome Foundation and all of the other butterflies out there. And we named our study after the symbol for the Dravet Syndrome Foundation as well. And this is a study looking, as I just mentioned, in all of those what we call comorbidities, those other problems besides seizures that children with Dravet syndrome have. And some of you have already contributed in some other studies, perhaps, to answer some questions. And those are all very important information to have. But what we're doing in this study is we're looking at children starting at one point and looking over 2 years in children to follow what the course of all of these effects are here that are listed. So that really hasn't been done before for people with Dravet syndrome, to look in the number of children who we will be looking in and for the length of time in what's called a prospective study. So that means we start, we ask questions, and then we follow children over time. There have been other times when, as some of you have, as I said, have answered questions about what's happened in the past or what's happening right now. But this is a way to take what's happening now and look over time in the future to see how these different issues change. And also importantly for us, what's the best way to measure these? Because right now we don't really have a good way to know what's the best way to measure walking issues in people with Dravet syndrome. What's the best way to assess sleep problems, or especially the cognition changes that happen in people with Dravet syndrome. So that's going to be a main part of the study, is to see what are the best tests to use. So that study is going on right now in the United States. We have many centers that are enrolling patients, and we've actually been very happy with the level of interest, but we are still looking for more patients, more people to enroll in this study. As I said, it's for children 2 to 18 years of age who have diagnosis of Dravet syndrome and have the SCN1A gene that's been shown to be mutated. So we encourage you to look for sites that are enrolling patients, and the Dravet Syndrome Foundation has further information, and I can provide it to you as well. I'm going to spend now the rest of the time talking about a very important study that we've just started. And continuing in the butterfly theme, we call it MONARCH. It's also a very beautiful butterfly, so it's a nice thing to put on all of our materials. MONARCH is a study where we are actually going to be testing in children our medicine, which is what we call the STK-001. It's what's called phase 1/2A study. What that means is this is the first time it's being tested ever in people. That's the phase 1 part. And the phase 2 part means it's not only being tested in people, but it's actually being tested in people with the disease. So a typical phase 1 study is done in healthy volunteers. And a typical phase 2 study is being done in patients. And because you're in a hurry and we're in a hurry, and we want to get information about this medicine soon, we've decided to combine those two and start right away testing our medicine in people with Dravet syndrome itself. It's called ascending dose study because the way the study is designed is we'll start at a low level, and then we will test at that low level in several children, make sure that everything is going well, and then we go to a next higher level. So we increase the dose. We do the same thing. We test in a small number of children, see if it's tolerated well then, and then we go to a next higher dose. So it's a sequential increasing of dose just to see how well the medicine is being taken up and handled by the body, and to make sure that there are no safety issues that show up. So the study is now actually enrolling. We just announced earlier this week that last week we had our first patient enrolled. It was a person who was between 13 and 18 years of age, and I'll explain to you why that was. But we are very happy now to tell you that we are actually we have introduced our medicine into people, and we are now continuing to have additional people who we will start giving the medicine to. So this study is what's called open label. It means that there's no placebo, there's no blinding of what the medicine is. Everybody enrolled in the trial we know is getting the actual active drug. And it is for children with Dravet syndrome, so 2 to 18 years of age. Sometimes I don't call a 18-year-old a child anymore, but it's a pediatric population. And we may enroll up to 40 patients. That would be the top number we would enroll. We may not get to that full number depending on how the study is run. And it's being run, as I said, just in the United States. We have about 20 sites that are already interested in our opening up to enroll patients. The way the study is designed, what we call a cohort, each group of patients who get a certain level of the dose of the medicine will be divided in two. There'll be two 13 to 18-year-olds who get the medicine. If they handle the medicine well, then we give it to 2 children 2 to 12 years of age. And then we look at all of that, and if there are no problems with the dosing in those people, then we go on to the next higher dose level, as I just mentioned. There is, if we need to, we can include an additional 3 patients in each dose level. So that's why when I told you about how many patients are going to be total in the study, we have several different factors that will determine ultimately how many children we can enroll in the study. And so we can't give an exact number. But for each cohort, as I said, the initial plan is to do 4 per cohort before we go from one to the next. The plan right now is the study is divided into 2 parts. The first part, part A, we will look at 2 dosing cohorts. As I said, the lowest dose cohort is already started, and we're going to be dosing the additional 3 patients in that cohort very soon, we hope. And then we will go on to the second cohort, the next higher dose. To go to higher doses above that, when we submitted our application to the FDA for this study, they asked us to go back and do a little bit more testing in animals, because when we did our pivotal trials, when we looked at the way animal testing is done for safety for medicines, we test up to a certain level and we see if there are any safety problems. We went in our pivotal trials in the animal testing, we went to a very high level and we did not see any safety issues that prevented us from going into the clinic. But the FDA said, well, we want you to go even higher. We want you to to test higher levels and see, is there any safety issue that shows up that we might have to look out for? And they said, we want you to come back with that information before you go into the higher doses. So that's what we're doing right now. We're collecting additional animal information at the higher doses, and we're going to send that to the FDA and ask them then to allow us to dose at higher levels. So that's the current plan right now for the study. Why are we doing this study? As I mentioned, the primary reason to do the study is to look at how well the medicine is handled in the body, and that's what we call safety and tolerability. So to make sure that the medicine is safe. We also look at something called pharmacokinetics. So that means when the medicine is injected into the body, what does the body do with that medicine? How quickly does it break it down? Does it store it? Does it change it into a different form? So we'll be looking at that by taking some blood samples and by testing the spinal fluid as well. We will look also at seizure levels in the people in the study. We will do that by using a seizure diary. So every family of a child in the study will be recording every day how many seizures the child has. We also will be asking the family and their physician, the clinician, how is your child doing? How is the child doing? That's sort of an overall clinical status. And then we do a questionnaire on what's called quality of life that asks specific questions about how is this disease having an impact on the child with the disease. So these are all the things that we're looking at in the study. When you ask, well, who can qualify for the study? Who's allowed in the study? This is what we call our inclusion and exclusion criteria. So the inclusion criteria are the things that any participant must have. And the first and most important is that we ask for what's called informed consent. We explain in great detail exactly what we expect to happen in the study, all the different testing that's going to be done, and all the risks that could happen to taking this medicine. That's the very first thing and the very most important thing that we do in this study. And once we have explained everything and the parents have agreed that they understand all the risks and the potential benefits and all the parts of the study that are going to happen, and they sign a form of consent, then we continue with the study. As I mentioned before, children 2 to 18 years of age will be qualified to enter the study. All have to have the diagnosis of Dravet syndrome, and we're going to actually check that by asking exactly how the disease has shown up in a child and make sure that it is consistent with what everybody agrees is Dravet syndrome. And then we will also be testing all the potential entrants into the study, looking at the SCN1A gene to make sure that they have a change in the SCN1A gene that would be consistent with Dravet syndrome. In addition, the potential qualified people for the study will have to have had at least 2 treatments in the past and have failed those treatments, either because they didn't work well enough or there was some safety issue. And they have to be on at least 1 medicine at the time that they're entering into the study. And that has to be stable for at least 4 weeks beforehand. And in fact, any medicine or treatment that's being used for Dravet syndrome has to be stable for at least 4 weeks prior to screening. And that's an important thing because we know that changes have to be made sometimes to the treatment regimen, but we also want to have as much of a stability before entering the trial as possible so that we're not in the trial measuring some effect of a change in the treatment regimen. So that's why we have this 4-week requirement in there. So those are most of the key things. There are some other laboratory tests and a few other things that get tested, but these are the major must-haves for the study. What I call the exclusion criteria. what are not allowed. So if someone has one of these things, then they would not be eligible for the trial. And one would be is if they have a mutation in another gene that's also known to cause epilepsy. And that typically those are, if they can be in different forms, sometimes they have to be on both genes, so it's called the homozygous mutation. So we look at all of those when we do the genetic testing to make sure that there may not be another genetic cause for the epilepsy, which our medicine would not be able to treat. Sorry. There are also some medicines called sodium channel blockers, which we would not allow in the trial, especially because our medicine is trying to increase the total number of sodium channels. And if at the same time, a person is taking medicine that blocks those channels, then that would sort of be counterproductive to what we're trying to do. There may be other medical conditions going on at the time in the child that are unstable and could interfere with our ability to measure how our medicine is doing. So in those cases, and that would be primarily up to the clinician, the investigator who was assessing the child at the time, to make sure that there's nothing else that might interfere with the ability to do the study. And they can't be sick with a virus or some other illness right before the screening period, so that they're not coming off of some sickness otherwise that might interfere with our ability to know exactly what the baseline levels are. And then if there are other issues that may affect our ability to do study, those are also not allowed. So that's the list of the major reasons someone may not be qualified. Now, the other thing that I want to mention is these are the ways to find out more about the study. There is a website, monarchstudy.com. There is also our email address, monarch@IQVIA.com. And then the Dravet Syndrome Foundation has been a very nice partner with us to help spread information about the study as well. I will tell you that the Dravet Syndrome Foundation has done such a good job about spreading the news about this study that we actually have already by now a relatively long list of people who are interested in the study. Not all, we don't know whether all of them meet all those criteria that I just listed in terms of being allowed in to the study. But as you saw before, these are small groups of 4 patients at a time who we are enrolling. So overall, in terms of study size, this is considered a relatively small study. So when you combine a small study with a long list of people who are interested, then there is a possibility that we may not have an opening for every child, every family who wants to enroll in the study. And we know there's been a tremendous amount of interest. We know that there is a tremendous desire by families, parents especially, of children with Dravet syndrome to try medicines that may help the child more. And believe me, we understand that, and we are as eager as anyone to try to accommodate all the patients who are interested in the study. But there may be limits. So some of you who contact us through any one of these different ways to contact us, at some point may be told the study is full. And that may be very disappointing. It may not happen, but if you get that news, we understand it may be very disappointing. And we're going to do our best to try to make the medicine available to as many children as possible as we go forward. There is a team at Stoke. We all dressed up with the Monarch costumes here one day. This is actually Halloween. So you see here the other members of our team who are actively working to help enroll patients and make sure that the study goes as well as possible. Some of them are actually on the phone right now, but you see Nancy and Shar and Melanie and Javier in that picture as well as me. And my thanks go to them, and I'm sure for all of you who are going to be able to participate in the study, thanks go to them for helping out. So that's really the end of my presentation, just to show you we're we're fortunate to have Nicholas, who has Dravet syndrome, come and visit us in the company. And for us, it was very meaningful to have that interaction with both Nicholas and his parents. And you see in the middle there, one of our scientists, Anne, who was presenting some of the data that Laurie Isom may present at a future moment. So with that, I'm going to end the presentation and turn it over to Misty to go on to the next step here.
Perfect. Thank you. A lot of good information. I know everyone in the community is really, really excited. Thank you for being here and presenting, and thank you for taking the time to answer some questions. I just want to remind everyone, we do have in the chat quite a few questions I'm going to go through. If you want to ask something live, you may take yourself off mute and do that. We'll start here. In the primate study, there was no or only marginal change in the protein levels of the low dose group. Does that mean that phase 1 won't necessarily have any effect either because it's also a low dose?
So that is based on the data, again, that we presented at the American Epilepsy Society. And in our monkey study, what we did show was that many parts of the brain, even in the lower dose with that monkey study, many parts did have the twofold change in the NaV1.1 level that we were looking for. That study is actually quite difficult to do because we're comparing from one animal to another, and we're comparing at different times. And there are also some parts of the brain that do not make very much of the NaV1.1. We know that NaV1.1 is important for many different cell types in the brain and in many different regions, but the NaV1.1 is made at different amounts in different parts of the brain. So in some parts, it's easier to measure a change than in others. So those were some of the complexities in that study that made full detection not as easy. And so that is why in some of the regions, as the question is correct, there weren't quite the twofold changes that we were expecting. The other thing to note is that was at a relatively low level of the medicine. We have given higher levels of the medicine, and ultimately we will be giving those levels that we think are appropriate in the MONARCH study to be able to have the effect in restoring the protein levels back to normal that we want.
Okay, and another question was attached onto that. It says, can patients in the MONARCH phase 1 trial continue getting STK-001 and at the higher dose if that proved to be more effective after completing the phase 1 study?
That's a very important question. And the answer to that is yes. So we are intending to allow for what's called an open label extension study. So that means that there will be another study to follow MONARCH so that all participants in MONARCH will, if they meet certain criteria, and that's a big if, but if they meet certain criteria, will be allowed to continue getting additional doses, and those doses will be adjusted once we learn more about the medicine to a level that we think is appropriate. But again, that study, we have not filed that study. We have not gotten approval to do that study, so I can't promise that study will happen, but our full intent is, if things go the way we want, that study will be available, and children and patients in the MONARCH study will be able to continue getting the medicine.
Okay, and then are you also testing in healthy individuals and then it says phase 1?
The answer to that is no. So we are starting directly in patients with Dravet syndrome, and there's no plan to do any testing in healthy people.
Okay. Okay, so your publications mentioned that no obvious adverse events were observed in the mouse trials. What do you consider adverse, and are there any known signs or symptoms?
So there are several questions there. What we consider adverse, that again really depends on what we see. We look at many different things when we do our testing in the animals. We look under a microscope at the actual cells to see, is there any change in the cells? Is there any sign that the cells are having any bad effects? Is there any response from the body in those cells that would be considered abnormal? So we look at even down to the microscope level. And then we also look at how the animals are behaving. Are they eating properly? Are they gaining weight normally? Are they interacting with the other animals normally? Are they sleeping normally? So those are all the things, and we sort of divide those up into the what we call the clinical or the obvious findings, and then the microscopic findings. And when we looked in the animals, we did not see changes on either of those sides. So that's the degree to which we would call something adverse if we saw anything out of the normal there, and we did not see that. And Misty, remind me the second half of the question.
Are there any known signs or symptoms?
So to this point, as I said, there aren't any specific signs that we know of a bad response to our medicine. This type of medicine with these ASOs, these antisense oligonucleotides, have been given for other diseases as well. The most notable one being a disease called SMA, and that is a medicine called Spinraza, which has a very similar chemistry to our medicine. And so we can look to that medicine to see what are the effects of that medicine that might be related, that we may expect to happen with our medicine, although each medicine is a little bit different. And there are no major issues that have been found with Spinraza either. There are some other antisense oligonucleotides that have caused, when they've been given to the whole body, not just in the spinal fluid, but to the whole body. They have had some problems with kidneys or with liver or with parts of the blood. And so those are important things that we are going to be looking out for in our study. We have not seen those to date in any of the testing we've done, but since those are potential signs, those are things that we'll be really carefully monitoring for in our clinical trial.
Okay. Approximately when do you anticipate being able to enroll younger children, like around 2 to 12? And will this vary by site? Along with that, does each site have to enroll 2 older children first before enrolling younger children?
So the way the study is designed, it's overall in the study. So any site can, once the sites have enrolled 2 older patients, and it doesn't matter which sites do it, it does not have to be at the same site. But across all the sites, once 2 children, older children have been enrolled, then the 2 younger children can be enrolled. And that's the case for each cohort. So the 2 younger children, the 2 to 12-year-olds, will be enrolled as soon as the 2 13 to 18-year-olds have finished their dosing and have shown that they've tolerated the medicine well.
So I'm going to add on to this. Would you suggest that someone that may have a child in the age group of 2 to 12 to go ahead and get on a wait list, or do you think we should wait for that process?
No, certainly go ahead and pursue interest in the trial. We already have on the wait list, we already have quite a few children 2 to 12 years of age. So we are already allowing the investigators to, and in fact, because of the way the study is designed, we want to have 2 to 12-year-olds ready to receive the medicine. So
okay.
certainly I would encourage that, yeah.
Okay, and then how were dose amounts initially determined?
That's done actually for many studies that are just starting to test the medicine in people that have not been tested before. There's a pretty standard way that this is done for most cases, and that is once, as I mentioned to you, once from the animal testing, we know what a level is that was safe and well tolerated in the animals. Then we take a certain factor, and in most cases it's 10 times lower. So the starting dose is typically 10 times lower than what would be the human, what's called the human equivalent dose of that study, of that medicine from the animal study. So we take the animal, we know what the highest, best tolerated dose in the animal is. We figure out what is the equivalent of that for a human being. And then we have that number, we divide that by 10. And that would be the place where we would start, or even lower than that. So that's a typical way that a starting dose is chosen for this type of trial, and that's similar to what exactly we've done.
Okay. Have you published PK data for your mouse and primate studies, as well as immunoresponse to repeat doses?
So we do have one publication that I'll mention right now that came out just a few weeks ago in Nature Communication, which takes that whole TANGO mechanism that I showed you and explains it in more detail and in better pictures and with other targets as well. So that's Nature Communication. We do have another paper that's coming out shortly, possibly in the next month or so, that will share all of the animal data that we think is relevant. And so those data hopefully will be coming out, and all that information will be coming out soon in a written form for everyone to look over in detail.
Okay. A press release mentioned that in the mouse model that received STK-001 on postnatal day 2, 76% of mice were seizure-free over a 24-day period, while the rest had 1 or more seizures. I didn't see any graphs about the seizure frequency of the mouse model that got the drug injection on postnatal day 14. How did those results look?
So those results were looking actually just at survival in the animal. So how many of the animals died and how many animals survived. And that was looking at the P2 injection. And then we also did what was called a P14 injection. So in this mouse model, the mice typically start to have the seizures and start to die around 14 days of life. And so we did the testing of our medicine at that point as well. And what we did show, again, was that many fewer of the mice who got our medicine died than the ones who got just a placebo or what we call the control group. So there was, even at a later stage around the time when the disease was really manifest in the mice, there was still an effect of the medicine in protecting the animals from dying. We did not do any injections after that because the mice become more fragile to work with. But this to us was very encouraging that we know that even when the medicine is given around the time that the disease is very active, it's still able to have an effect.
Okay. How will it be determined that an individual needs a repeat injection? For example, if a patient gets an injection in January, will there be a CSF collection in 3 months to determine if injection needs to be repeated or if it can wait another 3 months?
Well, we will do that measurement in this MONARCH study, and we will be looking, as I said, in the blood and in the CSF exactly. And that will give us already quite a bit of information to know how many people are handling the medicine. And so in the future, there will not be a need for everyone to have that sort of measurement done. We will be able to make an estimate based on the information that we get from this first trial, how often we think it needs to be given. And so we will set an interval based on the information that we get and comparing that to the information that we saw in the animals. So in general, in the future, if the medicine becomes available to everybody, there will not be a need for everyone to have blood testing or CSF testing in order to know. We will set the interval already in advance, and that will be given primarily based on what we have seen in the early studies. There is a chance that there may be a way to measure the seizure response as well to determine that, but that's not the way we are intending to set the interval. It will be based mostly on how the body is handling the medicine.
Okay, so how will Stoke decide from all the names that could qualify who gets in and who doesn't?
Well, let me clarify. First of all, we don't decide. So as a company, we set the guidelines for who we think should enroll in the trial. But then all of our wonderful collaborators, the different neurologists, pediatric neurologists who are helping us with our trial, they are the ones who identify patients or who are contacted by families, and they are the ones who put them on the list. They are the ones who review all the results of the testing to say, yes, this child qualifies or not. And the way that will be done is mostly there is a list already with an order of patients, and we'll go down the list. So the group that is managing the study for us, we're not doing the actual management, but what's called a CRO manages the study. They will help go down the list, and when each child is reached in the list, they will notify the investigator and say, the child that you've listed is now next on the list. You can proceed with testing on that child. So it's really not up to us at all. We have no influence, and we cannot say this child should be taken before this one or affect anyone on the list at all.
Okay, so Dravet mice that received STK-001 on postnatal day 2 versus day 14, disease onset. The survival rate dropped from 99% to 85%. Is it therefore correct to conclude that as the Dravet patient gets older, STK-001 will be less effective?
Well, that could be one way to look at it. Now, the challenge here is that, first of all, we're talking about mice. And having looked at many, many medicines that have been tested in mice myself, and knowing that there have been thousands and thousands of medicines that have been tested in mice, it's difficult sometimes to say what happens in a mouse is going to be what happens in humans. It's very difficult sometimes to predict. Also, this mouse model is a very severe form of the disease. So, the fact that so many of the mice are dying and so many have a very high rate of seizures is not typically what's seen with children with Dravet syndrome. So in this case, it's a very severe disease, and Lori Isom, who will be telling you about the story as well, herself will tell you how encouraged she was to see this level of effect, knowing how advanced the disease is in these mice. To see any effect to her was very, very encouraging. And so to know whether this tells us whether the medicine will work in older patients is really almost impossible for us to know. The only way for us to know, the best way for us to know at this point, of course, is to test the medicine in older children and see what the response is. And that's fully what we're intending to do.
Do you have time for some more questions? There's some more on here, and I don't know what your schedule will allow, so I wanted to ask you.
Well, I'm happy to give, can we do it 5 more minutes?
Yeah, we can.
Okay.
So, I want to ask them in order as they were put in, so that's kind of what I'm doing here.
Okay.
Will Canadian patients seen by a doctor in the USA be allowed in the trial?
Well, at this point, there's no restriction. It's very challenging for someone outside the United States, especially in these days of COVID, to even come into the country, of course. And then there are multiple visits that happen. It's not just a one-time meeting with the investigator. There are multiple visits that have to happen. So overall, we found that people living in the United States who are nearby the sites have the easiest time, and even some who are somewhat distant from the sites can manage this, but coming from a different country can sometimes be a big challenge in terms of being able to meet all the criteria and all the necessary visits for the study. So we're not discouraging it, but at this point, it's a challenge. I can tell you that we're hoping, if things go well, we're certainly hoping to be able to bring our trial to Canadian patients as well as others in other parts of the world. So it may mean waiting a little while, which is a difficult thing for us to ask. We hate asking anyone to wait, but waiting might be the only option that we have at this point.
Okay. Can you give us a sense of potential timelines for future studies and the earliest the drug could receive FDA approval under like the best case scenario?
Yeah, many people ask that, and my answer is pretty much the same. There are so many different things that have to go into testing of a medicine that trying to predict these things is very difficult. You know, we have to first see, just in this very first study, how things go. We don't, as I showed you, we don't even know how high we can do the testing right now. We don't know how many dose levels we will be able to go to. We then have to, after we test single doses, then we test several doses together. And then we look at all those data, then we have to talk to, all over the world, we have to talk to regulatory authorities, people who control medicine availability, to see if they think that we can test our medicine in their country. And then we have to do testing on a larger number of patients. Could be tens and tens of patients, even 100 or more patients. We have to do the testing in that level as well. And then if everything goes well and we have the results that we're looking for, then we would file around the world to see if then we can make our medicine available to everybody. So just in the time that I had to take to explain all that, you can understand all the complexities. And so to be able to put a timeline on this is very difficult. It's certainly several years, you know, at least 2, 3, 4, 5 years. That's the range that would be typical for this sort of a medicine and this approach, but it could be longer. It's not going to be shorter.
I think we have time for just one more, and I kind of went through and saw this question listed a couple of times, so I wanted to ask it. Are eligible participants required to have specific seizure types or like a certain amount of seizures per month?
So yes, the answer is yes. There are what we call the convulsive seizures. So the ones that can be easily counted. Those are the ones that we're using to judge for entry into the trial. So I know many of your children have different types of seizures, especially absence seizures or myoclonic seizures, and those are more difficult to count and have a good way to record exactly that they're happening. So we are looking more for the tonic-clonic type seizures, the ones that are what we call the convulsive type, and so those would be the type that would be used for entry. That doesn't mean that if your child has the other types, they would be excluded, and it doesn't mean that the convulsive types are the only type that are allowed that would be measured in the study. But in order to know the minimum number, and there is a minimum number which would be discussed with the investigators when you're able to talk to the investigator about the study, they will go through all of that to tell you what the minimum number would be. Perfect.
Well, I know we have gone over time just to have you here and answer some more questions. We are very appreciative of everything that you were able to share with us today. I want to remind to everyone really quick, if you haven't registered for next Thursday, August 20th, educational webinar, you should go ahead and do that. It is going to be living with seizure clusters, how a rescue medication may be able to help. So we hope to see you then. Dr. Tico, anything else?
Well, again, I want to thank the Dravet Syndrome Foundation for hosting this and for being willing to let us share some information about our trial. I want to thank everybody who's shown an interest in our medicine. And I want to, again, tell you that if you have other questions, and I know that I try to answer as many of them as possible, but there are always more questions. We're happy to answer them, either contacting us directly at the company or those other ways that I showed you on the previous slide.
Perfect. And this will be recorded, or it has been recorded, so we'll post it on the website. Definitely, I believe it's by tomorrow. So you can always go back and find the email address there that Dr. Tico gave you. Other than that, I hope you guys have a good Friday, and I'll see you next week. Thank you, Dr. Tico.
Thank you. Thank you, Misty. Thank you, Jenny. Thank you, everybody.
Thanks.
Thank you.